welcome back to Biology with Gracie. So today I'm going to be doing my third video on carbohydrates for A-level biology. So in today's video we're going to be talking about biochemical tests. So the first ones we're going to be talking about are tests for sugar. So we're going to be talking about the Benedict's reagent, which is used to test for reducing sugars, and how it's also used to test for non-reducing sugars. We're then going to go on to talk about the Starch test, and we're going to be talking about the iodine potassium iodide. So there are two main types of biochemical tests. So we've got Benedict's reagent, which tests for reducing and non-reducing sugars, and then we've got the iodine potassium iodide, which is a chemical test for starch. So Benedict's reagent is an alkaline solution of copper 2 sulfate, and it can be used to test for the presence of reducing sugars. So some disaccharides and all monosaccharides are reducing sugars, and what they do is they donate an electron to the Benedict's reagent. So the method that we use to test for reducing sugars using Benedict's reagent is as follows. So the first thing you need to do is add 2 centimeters cubed of the food sample you're testing to a test tube, and this needs to be in liquid form. You then need to add 2 centimeters cubed of Benedict's reagent, and then you want to heat the mixture gently in a water bath for 5 minutes. And if the solution turns brick red or sort of an orange brown colour, this means that there's a reducing sugar present and it is a positive result. So this red precipitate is insoluble and it is actually copper 1 oxide. So Benedict's reagent can also be used to test for non-reducing sugars. So all polysaccharides and some disaccharides are non-reducing sugars. So the test for non-reducing sugars using Benedict's reagent is slightly more complex than testing for reducing sugars, but nevertheless it's still fairly simple to follow. So the first step, you've got to perform the reducing sugar test essentially. So go through the steps that I talked about previously when testing for reducing sugars. And if there's no colour change, that means that there is no reducing sugar present. So we know that it is not a reducing sugar. So now we can go on to test whether it's a non-reducing sugar. So what you need to do is add 2 centimeters cubed of your food sample, again in liquid form, to a test tube, and then add dilute hydrochloric acid. You then want to put this test tube in a water bath for 5 minutes, and what is hopefully going to occur is that the dilute hydrochloric acid is going to hydrolyze the disaccharides and polysaccharides into monosaccharides. So then sodium hydrogen carbonate is added, and this is to neutralize the mixture, and this is because Benedict's reagent actually doesn't work in acidic conditions. You're going to retest the solution by adding your 2 centimeters cubed of Benedict's reagent and place in the water bath for another 5 minutes. And then if a non-reducing sugar is present, there's going to be a colour change from blue to that brick red orange brown colour once again. So the second test is iodine and potassium iodide, and this is a chemical test for starch. So what happens when you perform this test is the solution turns from an orange brown colour to a blue black, and this therefore shows that starch is present. So I hope you found this video very useful and helpful, and I hope it's given you a bit more of an insight into biochemical tests. Please make sure to give it a big thumbs up if you have found it useful, subscribe down below, and make sure to follow me at Biology with Gracie on Instagram, and I look forward to seeing you in my next video. Bye guys!